I am so delighted to be joined today by Professor Simon Walker-Samuel, who is Professor of Biophysics at UCL. It's great to see you again, Simon. We had the pleasure of working together just a couple of months ago. You came for a very interesting discussion with Dennis Alexander at one of my schools.
We did. That was great, yeah.
That was a fantastic discussion. In fact, I'll definitely put it in the link. You're talking about genetic determinism, and it was sort of everything I hoped it would be in terms of inspiring students and staff. You've got a
Good.
very interesting background. I can't wait to get into it.
Yeah, very happy to. Yeah, it was fun chatting with Dennis. He's really interesting.
Yeah, we'll definitely try and bring him on here. So, for those of you who don't know, those who don't know anything about biophysics, could you just tell us a little bit about what it is that you do?
Yep. So my background's in physics, which is the physics bit of the biophysics, but applied now to biology. So I did a undergraduate degree in physics. So was just studied uh, just purely physics from that perspective. Then during that time, I spent a year working at the Institute of Cancer Research, which is in South London kind of area, Surrey, and applying sort of what I'd learned in physics to studying MRI scans, so magnetic resonance imaging, which is a very physicsy thing. So most hospitals nowadays have an MRI scanner, and that relies on all sorts of interesting physics like quantum mechanics and superconductors, along with a lot of kind of other sort of more conventional classical stuff. So that's kind of where the physics came into medicine and biology, and trying to use understandings of physics to image inside people. So to look at people's tumors, if we suspect someone has a cancerous tumor, then we can use MRI scans to look inside them. So that really heavily depends on the physics. But then that kind of feeds into other areas, which is sort of more my focus now, which is using physics to understand certain characteristics of tumors. So looking at things like blood flow. So using things like fluid dynamics, which is again a very sort of mathematical physicsy discipline. So trying to understand things like drug delivery to tumors. So if you inject a drug into a patient, understanding where it goes is actually not that straightforward.
No.
And they tend to go to places you don't want them to go. So it's that kind of using numerical, mathsy, physicsy stuff applied to medicine and biology. So that's the biophysics that I do.
And there's obviously big implications there as to how the medicine is delivered, whether it's injected or taken orally or all
Yeah.
sorts.
Yeah. I mean, in terms of what we call modeling, so sort of forming mathematical models to try and understand quantitatively. So, we want to know exactly how much of a drug is going to get into a person's tumor. And the reason for that is that anti-cancer drugs are usually quite toxic. So they have effects on normal tissues that you don't really want. So you want as little affecting the normal tissues and as much affecting the cancer as possible. And there's lots of ways that people have suggested for engineering that exact, situation where you get as much into the tumor as possible and there's little knock on. So injecting drugs is tricky for that reason, because when you inject something, it goes everywhere. So you can design drugs, for example, that you hope will just go to the tumor and really not touch the normal tissues as much. But equally, there's other forms of anti-cancer therapy. So not just injecting drugs, there's things like radiotherapy. And that's where you fire radio waves into a patient and try and basically kill off the tumor by cooking it, essentially,
Yeah.
using radiation. And that's all based on physics as well.
Well, while we're right here, I want to ask you if your work crosses over with cancer immunotherapy at all.
Yeah, it can do. So we've had a few projects trying to understand. So immunotherapy is typically something called T cell therapy these days, where you take these immune cells out of a patient and you sort of re-engineer them to attack tumors.
Yeah.
So the tricky thing about tumors is they are part of the person. They're not like an alien foreign body. They are actually just the sort of natural
Yeah.
processes in the body gone wrong. So if you can understand where T cells are going to go inside the body after you've re-injected them, and then how they do things like, are they active inside there? It is kind of sitting there waiting to do something, all those kind of phenomena. It's really useful to be able to study quantitatively. And physics is a really good way of kind of quantifying things. It's not the only way, but it's a pretty good way. So bringing that kind of numerical physics stuff is a really interesting. I was going to say it's a new avenue, but we've been doing it for quite a long time now.
Yeah.
So it's sort of establishing itself still, but it's still quite novel, but not completely.
Yes, and it has been around for a while, and yet so few students know they can go and study this. I sort of think of
Yeah,
biophysics as being as old as Francis Crick and George Gamow and all those people who
yeah,
came out of physics after World War II and transitioned to biology, who
yeah,
gave birth to the molecular biology revolution.
yeah,
Is
yeah,
that
yeah.
sort of how you see it? You're
It,
continuing
it,
that
it,
fine line.
I guess so. And you can get very kind of focused in on what you do and forget the kind of pedigree for it. But yeah, so Owen Schrödinger was a very influential physicist in quantum mechanics. And after he'd done a lot of the really interesting stuff in physics, he wrote a book called, I think it's called The Physics of Life. I may have got that wrong.
What is
But
life?
he was, what is life? Okay,
Yeah.
yes. Um, and yeah, so he kind of suddenly went, oh, I wonder if this is all applicable to biology in some way.
Hmm.
Um, and that kind of spawned a bit of a revolution that kind of has had peaks and troughs over time. Because
Yes.
it's hard. Life is really, really complex. So physics has tended to focus on really hard conceptual stuff, but which is fairly simple to model. Whereas life and complex biology really isn't that. So a lot of the time you don't really know what you're dealing with,
Hmm.
and that makes it difficult. So that's why what we do a lot of the time draws on imaging. So trying to sort of understand the structure of complex objects. And tumors are often incredibly complex because tumors don't obey a lot of the normal rules that biology forms to make it kind of cooperate together and make sure that we can continue our life. Essentially, tumors are kind of where those rules have been switched off to an extent, and as a result, you just get this incredible complexity forming that is then quite hard to model unless you can really sort of get a fine handle on that structure and then apply physics on top of it.
Gosh, there's already a million things that we brought up that I'd love to get into more. But thinking about the long-held dream of humanity that medicine will be personalized and precision, you're dealing obviously with modeling very dynamic things and sometimes quite unique to the individual's own physiology. So you're modeling tumors, you're modeling as sort of where the cancer is in the person, how advanced it is. You can't generalize all that much, can you? It needs to stay dynamic. How do you manage to achieve that?
Yes. So this idea of personalized therapy is kind of the dream. Um, and it's been the dream again for quite a long time. Yeah, because um, tumors are fairly unique. I mean, so personalized medicine kind of cuts across lots of different areas of medicine, not just cancer, but it's really the case for cancer because you could have a cancer that doesn't respond to a particular treatment, or you can have a cancer that does respond to that treatment, and it's often not particularly well known why one does and one doesn't. So there's a scientific area of inquiry there. Some of that kind of touches on physics. So, for example, some tumors have a lot of blood vessels in them, which means if you inject a drug, it's more easy to get into them. Is the sort of naive hope sometimes that
Mm.
more blood vessels equals more delivery means more killing the tumor with a drug. It doesn't always kind of translate to that for all sorts of other reasons. So, really understanding each person's individual tumor is exactly that dream. Can we get enough data and enough information on each person's tumor that we'll be able to then treat it as an individual rather than how medicine currently works, which is okay, you've got this type of tumor, we always give this kind of drug. If you respond, great. If you don't, we'll try the next one. And if you don't
Mm.
respond to that, we'll try the next one. And so it's always kind of, and you're always building up a picture through failed treatments in that way.
Mm.
Whereas it'd be much better to go, we can see exactly what will work in you.
Yeah, it's sort of in terms of weaponry against cancer, it's like switching from grenades to sniper rifles. Like, can we
Yeah. Yes.
rather than just say you're a human with this cancer, therefore cut it, blast it, or burn it, the traditional means. You have your own unique physiology, and you've had your cancer in this place for this long. So we'd like to specifically and with as few side effects as possible hit this.
Yeah.
It's the dream. How far away do you think that dream is?
Oh,
Big
that's the
question.
classic question. It's the classic question. We always underestimate, unfortunately, how quickly things are going to become a reality.
Mm,
And you always like to say in 5 to 10 years at least, and sometimes it does actually do that. You have a sudden sort of acceleration and things happen. Other times you sort of it's just a piecemeal approach, and there's often it's not just the science that's the barriers. It's actually getting these things in place in a clinic with a suitably trained team that sort of understands it. Though, that's why, and regulatory bodies as well. You
yes.
have to really show that what you're doing works and it works well and you can evidence it. And all that takes a lot of time.
Mm,
That said, so what we do there's the area that we work in is often called digital twins. So you can take MRI images of a patient and you use that to get the structure, and then you try and build up a treatment plan or an understanding of that tumor that's individualized. And this is
mm,
sort of representation of the patient inside your computer. So this is sort of this idea of a digital twin. And then you can do experiments on that digital twin in a way that you wouldn't want to on the patient because it takes time and everything. So you could, in an ideal world, you could try out 100 different drugs on your digital twin and go, actually, I think that one's going to work best for this individual person because of these particular characteristics. That feels like it shouldn't be too far away. And particularly with things like AI coming in more and more into medical practice,
mm.
I think that should enable these things to happen faster. But even getting AI into the clinic is a challenge, and that's taking
Yeah.
longer than people might want. So yeah, I'd like to say 5 to 10 years, but you never quite know. And in the meantime, patients need it. It's something that's really critically needed. So if students want to get into a really useful area, I'd say this is a good one.
Yeah, yeah, you heard it here. Let's jump a bit more into the computational side because that is really disruptive. I mean, the Nobel Prize was awarded for AlphaFold last winter, um, and so the computational modeling side of what you do. I do quite a lot of homology modeling and bioinformatics in my work. Could you walk us through the computational side of what you do? Because it's the Centre for Computational Medicine that you run at UCL
Yeah,
now, isn't it?
it is. So we sort of have three things that we combine. So the idea is, so we've got imaging, which is things like MRI scans, PET scans, CT. I'm drawing a sort of Venn diagram here with my finger. So there's the imaging bit there. There's sort of computational modeling, which sort of brings in the biophysics and the fluid dynamics and the real kind of physics understanding of the world. Or biochemistry or genetics doesn't just have to be physics, but it tends to be what we focus on. So that's the kind of second bit. And then here we've got the AI bit, which is trying to leverage things to make that easier. And by easier, so for example, with the imaging, it's often quite hard, even if you've got a really amazing MRI scan and you say, okay, the human goes, okay, I can see there's a tumor there and I can kind of see some blood vessels inside it. Extracting those features and making them into a digital twin is surprisingly tricky. And it's something that we as humans can do really well and we can kind
Mm,
of draw around them. But for computers to do that, it's a lot harder. But AI, and I mean, that whole approach is called segmentation, image segmentation. AI is getting really good at doing that. They say, okay, there's the tumor, there's the person's organs around it, there's the blood vessels. So we can build that using AI into this sort of digital twin and then apply the kind of biophysics stuff to it. So trying to understand things like blood flow and drug delivery
mm,
on top of that. So those three things together, and of which the AI and the computational modeling are the kind of computery bits building on the data that you get from imaging. And you can also bring in other bits of data, so genetics, biopsy data, all those kind of things to build up this kind of multi-mode or multi-dimensional characterization of individual patients that you can then use to kind of define their treatment plan. That's the kind of dream pipeline.
It sounds incredible, and we love the interdisciplinary thing here. Big time. We love polymaths, people who've got skills and extra things. Could you tell us a little bit about how you got into biophysics? Because you did your
Yeah.
biophysics PhD, but did you have a medicine background or a physics background? And
No,
how did you
no, so
cross over?
it was physics. I mean, it was okay. Um, I can give you the honest answer, which is
Please do.
that it's sort of like it's almost sort of Brownian motion diffusion, how you often end up in these things. I'm very glad I ended up in the area that I'm in, but I did an undergraduate physics degree, and it was an MPhys program, so Masters in Physics, which is amazing. I had the best time. And again, for the any students watching, doing a physics degree is incredible because you learn about the universe in a way that you just don't at GCSE or an A level.
Hmm.
So you look at, quantum mechanics and general relativity and things like that, and it is absolutely mind blowing. So I had the best time.
Where did you
Um,
do that, by the
I,
way?
so that was at
Which,
Surrey
sorry.
University. Yep. Um, so and learned a bit about particle physics. And they had a great link at the time with CERN, which is the particle accelerator. So they were
Wow.
building the Large Hadron Collider at the time.
Wow.
And they had loads of projects doing that. And I was like, oh, yeah, that sounds amazing. I want to go and build the Large Hadron Collider, build magnets and all that kind of stuff. Um, that fell through for all sorts of different reasons. And a great lecturer at the time, Simon Doran, said, instead you can go to the Institute of Cancer Research to spend a year there working on MRI scans. And I thought, okay, yeah, that sounds good. That sounds quite a worthy thing to do, looking at cancer rather than this thing. So tried that and just got the bug. So complete chance, really.
Wow.
Part engineer. I was doing a physics degree already, so I was halfway there, but it's slightly chance based, and loved doing that. That fed into a PhD with a guy called David Collins and Martin Leach, who ran the MRI department there. And then you just it kind of snowballs from that point onwards. So and you go, okay, I've got a PhD now. I should do a postdoc position, which is where you're kind of working as a scientist after the PhD, building on the skills that you've learned during the PhD. So I did that for 2 years with a guy called Simon Robinson at the Institute of Cancer Research, and then moved over to another postdoc position at UCL, and carried on doing it really. And it's often the case, just when you get bit by this random thing and you kind of enjoy it and it kind of becomes part of you somehow, and then you can't let it go. And I've been lucky enough to kind of carry on doing that for the last 20 years, I think, something like that.
Wow. And so,
Yeah.
was there any genetics or medicine or biomedicine that you had to do as part of your training?
Nothing formal. And it might be different now. There's probably a much better appreciation of interdisciplinarity and the need to have formal training. I mean, so I have a few colleagues. I know an oncologist, for example, who also has a degree in maths. Applied, I think it's maths applied to physics. We had this discussion the other day, something like that. But
Wow.
so they've got both. He is a practicing oncologist with a medical degree and a physics degree. Maybe overdoing it. I don't know. For me, it was a lot kind of picking it up on the job, going to seminars for colleagues that do know about biochemistry and genetics, and you just gradually kind of get this. You build a bigger and bigger picture of the world. But I think fundamentally, starting out in physics, you tend to kind of view the world a certain way,
Hmm.
which is kind of numerical, and it's different from a formal training in biology. At least when I did it, I think you come out with a slightly different sense of the world, which you can adjust and mold and develop and everything. But I think it sets you out on a particular trajectory, which is slightly different. And you just sort of view problems in a slightly different way than if you've got a biology training. Both are great ways of looking at the world, but they are different. And I think they complement each other quite well. But I had to do quite a lot of reading to get up to speed with biology and medicine, having not done them since hadn't done biology since GCSE, I don't think, and then kind of thrown into this PhD, having to kind of talk about tumors and understand them. So
Yeah,
you just self-directed.
but it's a fantastic perspective to bring, and as we've already said, historically, it opened up biology in a whole new way. I think also Ramakrishnan, who won the Nobel Prize in 2009 for visualizing the ribosome, he had a physics PhD as well and sort of fell into biology because there was a problem that needed solving. So
Yeah.
we've mentioned it a few times, the Institute for Cancer Research. I keep wanting to abbreviate it to the ICR, but there
Yeah.
is
Yeah.
another ICR called the Institute for Creation Research, which is like a
Ah,
creationist place. And I don't want people thinking you have anything to
very
do with
different.
that.
Okay, I wasn't aware of the Institute for Creationist Research. For me, ICR is just the Institute of Cancer Research.
Yeah,
Whenever I say it, but yeah,
yeah, it's
it's
the first
probably a
thing.
common acronym,
Yeah, yeah, I wouldn't want anyone to get it wrong. Tell
right?
us a bit about the ICR and your time there and what they do. And
Yeah, it's a pretty amazing place. So they've got two sites. They've got one in sort of South London, Chelsea, Kensington kind of way, and they've got a bigger site in Sutton in Surrey, just south of Sutton, which is where I was based. And all they focus on, hence the name, is cancer research. So they have this incredible expertise in many different disciplines that all kind of come together in great ways. So it's a really interesting place to work. I was based at the MRI department. This may have all changed since I've been there, but they do lots of work on genetics and developing new drug targets, identifying new drug targets. Um, they do clinical work, particularly looking at things like prostate cancer, um, and they have had some successful trials looking at drugs. They're into immunotherapy at the moment, so a
yeah.
broad range of interest and really kind of impactful work. And they've had that for many years, so they are quite a powerhouse for doing, incredible, world-changing work in cancer.
Yeah, we're very privileged in this country to have some of the best facilities in the world, particularly in this area,
Yeah,
providing great opportunities for students to do fully funded programs.
yeah.
One thing that you do quite a lot of is tissue modeling. So I wondered if you could tell us a bit about that, what it is and how it works.
Right. So there's two types of tissue modeling we do, I'd say, maybe three, but I'll focus on two. So there's what they call in silico, which is kind of computer models, this digital twin idea, but also in vitro. They've got these weird Latin kind of names for these different kind of systems. So in silico means in silicon computing. In vitro means, I guess, in glass. So looking in petri dishes. And this is where you can take tumor cells. This is very kind of biology based. Take tumor cells and look at how they behave in slightly constrained, unnatural circumstances, but it's kind of often the best we've got. Um, and the third, maybe I'll touch on the third, is in vitro, which means in living tissue, where it's often, sort of animal experiments, looking at mice models and things. That's sort of becoming less and less popular, I'd say, because people don't really like doing, for ethical reasons generally, they don't like doing in vitro models so much. But there's still a really important role that they play in research. But the two we really like to focus on is in silico and in vitro. So the in vitro stuff is really coming on leaps and bounds. So, I mean, it was really back in, I guess, the 1950s where people realized you could take biopsies from people's tumors, so take out some of the cells and grow them up. And they found that particularly tumor cells were often, well, sometimes immortal. So they would just keep replicating over and over and over, which is kind of a characteristic of cancer itself, that you get this unconstrained, uncontrolled growth. Whereas normal cells have this sort of inbuilt mechanism where they go, right, I've divided a certain number of times. I'm going to stop now. Enough's enough. Whereas tumors kind of have the break often taken off, where they just kind of replicate, which is why you get this kind of mass of cells that constitutes a tumor. So you can take those cells, you can put them in some sort of dish with some sort of growth medium, so the things they need to survive, sugars and that kind of thing, and watch them grow. And you can see, okay, if I drop a drug on it, how does it respond to that drug? So this is kind of how the early sort of more interesting therapies were developed by using these kind of approaches. But they're now progressing beyond that kind of single layer, flat layer of cells, which is quite a kind of contrived situation, to actually growing them up in 3D.
Yeah.
And what they could, people have different names for them, but there's things like organoids or tumoroids, or there's always got oids on the end.
Yeah.
So this idea that it's a sort of three-dimensional growth that you're sort of simulating, and cells behave differently, it turns out, when they're flat to when they're in lumps. So, and it tends to be the lumpy scenario that's more representative of real tumors, because that's how they grow. And that's fine. So you can grow up these spheroids, organoids, whatever you want to call them, to a certain size, but very quickly they start lacking oxygen because oxygen can't get to the middle. And this can happen in normal tumors as well, that you, once they get to a certain size, they need to establish a blood supply. So you need to grow blood vessels in, and that's typically from surrounding normal tissues, that the tumor starts emitting growth, like calls for blood vessels to be grown towards them. There's a molecule called VEGF, for example. So when cells become really lacking in oxygen, they emit VEGF, and the surrounding blood vessels go, there's some hypoxia, some low oxygen going around here. Let's grow vessels in. So you start getting these kind of vessels forming. That has been really hard to kind of create a model of. So,
Yeah.
you can grow up the cells in 3D, but getting blood vessels until fairly recently has been quite hard. But suddenly we can do it. So this has been this sort of flurry of activity where actually you can, seed endothelial cells. I've got a student working on exactly this in my group. And he can actually, just sort of start growing blood vessels inside these spheroids and actually sort of then perfuse a flow material through them. So they actually make kind of tube-like structures in the way that blood vessels, real blood vessels are. So suddenly you're like, oh, wow, this is a kind of, this is a step change. Like I was saying before, you never quite know what's going to happen to make this kind of step change where you go, now we can do that. This means that we can really simulate the tumor growth in a way that we couldn't before. And then coupling that with the in silico stuff, so we can image these in vitro models and then make models of them in the computer and then use that to understand tumors inside humans. That's the kind of next step stuff.
Do you ever envision that the computational methods, which are advancing so much, could ever fully simulate the tumor environment and replace large sort of lab-based drug discovery processes completely?
Um, I d- so cells are kind of black boxes that you can. The reason that so people are making incredible in silico, so in computer models of individual cells, and the way that they do that is they've prodded them and kind of worked out what happens to them under any number of situations. And I'm simplifying, sort of the whole biochemistry of cells is really well understood. So, how they function internally has been studied for, over 100 years. And you can kind of encapsulate all of that knowledge inside a computer model now and sort of replicate the behavior of individual cells fairly accurately. But there's still a chance that a real cell won't behave in the way that you anticipate it will be, because maybe there's a gap in our knowledge and understanding from experiment of exactly how it works when you prod it in that way, or kind of introduce a new agent or something. So we're getting close to that point. But the vision that we work towards is the one of complexity. So you might have a kind of simple model of how the cell is working, but maybe we can simulate 1,000,000 of them or 100 million of them in consort, and then see what happens and how they kind of behave relative to each other. What happens when that region there is lacking oxygen and calling for blood vessels, but actually this region here isn't. Is that region going to respond differently to therapy than that region for some reason? Or radiotherapy, for example, for radiotherapy to work, you need lots of oxygen in it because it releases free radicals and kills, and damages the DNA and kills the cell. So if you can say, that region has got lots of oxygen because of some reason, and that region hasn't, it's going to respond differently. So In the sort of immediate future, it's still going to be fairly simplistic, but it just sort of gets more and more complete our understanding of how the cell works, how a cell works when it's next to a couple of other cells, how they work when they're sort of surrounded by 1,000 cells. So scaling it up to that level where you start having a system that's so complex that you can't understand it in an experiment because you've got so much going on and so many interactions. That's where I really think the modeling can come in to, is a really useful tool because you can really capture everything going on at any moment in a way that you just can't in an experiment.
Right. Well, in terms of treatment itself, do you envision a day where perhaps immunotherapy and maybe DNA repair-based treatments as well could completely replace chemotherapy, radiotherapy, and surgery? You know, the much more invasive ones that can damage a lot of healthy tissue as well.
I mean, you gotta hope so because I mean, chemotherapy has a reputation for being really brutal, and it really can be in a lot of people, and a lot of people really suffer as they go through it. Some people just whistle through it, and and they don't even say, oh, so they don't notice, but they don't become ill at all. So it does have this reputation for being brutal. That isn't always the case, but there are people who really do suffer the side effects of chemotherapies. So moving beyond that, and I mean, we're still using a lot of agents that were developed in the 50s that do the job, but they are quite harsh. So sort of less harsh treatments that are more targeted. So things like immunotherapy that are designed for the individual patient and to kind of not have these knock-on side effects. Yeah, that's got to be the future, and I think that's kind of where everyone's pulling, is kind of to move away into start defining the next generation, which is already here. I mean, I'm saying it's the future, but immunotherapy is actually happening. It's a thing.
Yeah, we just got to get the costs down. And the costs, I mean, in my mind, the costs will come down just because more people get into it, more research happens, more biotechs start producing the medicine. Is it as simple as that, do you think? Because, I mean, it costs about $500,000 per go per person at the moment. The NHS can't afford that.
No, and it, I if it follows the normal pattern of things, it will become cheaper. As people find easier ways to say, harvest and adapt the T cells. T cell therapy, which is where you have to take the cells out and re-engineer them, then put them back in again. That may not be the only way of doing it. There may be other mechanisms we can exploit that I don't know about. This is not my area of expertise before I start trying to explain it. But it may be that T cell therapy is the kind of first wave of immunotherapy, and there are other, technologies coming behind it that aren't so expensive. But you are absolutely right. Health economics is often the thing that stops progress or is a factor in the progression of new therapies. And I mean, and that cuts across lots of different disciplines. It's not just the medicines. It's a lot of other things that, if they're too expensive, they just don't happen, even if they work particularly well.
Let's get into a bit more about your group, the Centre for Computational Medicine at UCL. What sort of other things have you been working on recently, or are you working on now?
So cancer is our background. However, we also do a lot of work in retinas, which is the kind of the organ at the back of your eye that's responsible for vision, essentially. The reason we did that is kind of linked to cancer. So the reason we started looking at the retina as an interesting organ is partly because of the kind of imaging that you can do of the retina. So the eye naturally has a lens at the front, which is used to, taking light from the outside world and focusing it onto the retina. And the retina then encodes that into electricity and goes into your brain, and you can see. It also means you can do the opposite, that you can kind of shine a laser in through that lens and get amazing detail of the retina itself. And the retina has at the front or the inside an amazing blood vessel network. So we're interested in blood vessels for delivering material and looking at biophysics, and you can see it in incredible detail. And unfortunately, that blood vessel network is really sensitive to high glucose concentrations. So if you're diabetic, one of the complications can be that you lose a lot of the small vessels in your retina, and you start losing your vision. And one of the the endpoints, one of the sort of late stage clinical presentations is loss of vision when you've got diabetes. But also, when you start losing that blood vessel network, the cells become hypoxic. They start releasing VEGF, which is this molecule I was just saying about, which is released when cells haven't got enough oxygen. And the blood vessels respond. They become what's called angiogenic. They start sort of making this really rapidly formed blood vessel network, which is really badly, it doesn't have this lovely kind of branching network that the normal retina has. It has this sort of malformed structure, which is sort of very similar in many ways to tumors. So you can see this happening kind of in real time. And there's clinical data, loads of clinical data from ophthalmology clinics. So I've got a good colleague from Moorfields Eye Hospital, and who's an ophthalmologist, Ranjan Rajendram, who can get hold of amazing images of exactly these processes happening in a way that you really can't get in tumors, because they tend to be embedded inside organs, and MRI data is low resolution. It's really tricky, but you can see this exquisite detail, which is a kind of biophysics dream, because you can kind of model blood flow, and you can model angiogenesis, and you can see all these processes happening in real time in real patients. So there's an interesting angle looking at treatments for for diabetic retinopathy, which is this process of loss of vision. So trying to understand and model the eye, but also then relating that to cancer and sort of understanding some of the biophysical processes going on in that. So it's like a model system as much as anything. So we do that, and again, there's a sort of AI challenge in that, because you want to segment the blood vessel network, and so we train neural networks to do that. There's the computational biophysics, so again, it's that kind of three-pronged Venn diagram. So that's one thing. My colleague Becky Shipley works with nerve damage, so she's the co-director of the CCM.
Yes.
So yeah, it's kind of broad, but can always go back to these three things of biophysics, imaging, and AI.
Yeah. And what a whole world it's opened up, because you can do that for anything, really. You can apply it liberally across all of the life sciences and health. In
Yep.
thinking about how students and young people can get into this sort of thing, because it is an exciting, vibrant world opening up. Was young Simon doing a lot of model building? Obviously dreaming of being a physicist, not a doctor, but what were
Yeah,
your
yeah.
first steps? Were you just building a lot of Lego when you were a child? And
When I was a kid, I did play with a lot of Lego as a kid, actually. Uh, I did a lot of music. I don't know if they're linked. There seems to be a sort of link between physics and music. There's a lot of kind
yes,
of physicists who also are sort of, in my case, a wannabe musician. But so I was doing a lot of that. But coding as a kid, I did a bit on
mm.
the BBC microcomputer. What were they called? The first wave of home computers was confusingly made by the BBC, I think. I'm not quite sure what the link to the BBC was, but anyway, I had this. You could code in a language called BASIC, and so I did some of that, and then kind of didn't do a lot of it after that, but did sort of music engineering, so sort of producing music. And some sort of electronic stuff as I got a bit older. So I set up a studio with a friend, and we did sort of drum and bass and all this kind of stuff. So it sounds awkwardly toe curling at this point, but but it was kind of very technology based, the music that we were doing at the time. Um, and that was after I'd left college. So I took some time out, and we sort of did that for a bit. And at the same time, I was kind of reading about physics, and I was like, oh, actually, I really want to do a degree in this. So kind of went back and went to university at that point. But realizing the power of coding, I think, is what made a huge difference. And I really started picking that up in the undergraduate degree, learning an ancient language called Fortran that barely anyone's heard of these days. But that was a thing. I don't know why we didn't learn C++ or something, but yes, Fortran was the language. And then a couple of languages later, then started coding in Python, so which is just taken off. Everyone
Yeah.
uses Python now as a coding language, and you can do anything in Python. And AI is slightly changing the landscape a bit now. So generative models are changing the landscape, but up until maybe sort of 5 years ago, I could control the lights in my house using Python scripting and, you know, even code music up. So making, sort of electronic music, but through coding. And it just enables a whole new level of power
Mm.
over your world and your environment. So I definitely was evangelical about coding. Um, and now it's kind of been democratized in a whole new way, coding through large language models. So ChatGPT, for example, you can say, write me code that will do this thing, and it will generate a script for doing it. So you don't even have to really do the coding anymore.
Wow.
So whether coding is gonna will continue to be that key skill that enables, I don't know.
Mm.
And we're sort of entering this whole kind of brave new world of what is going to be the important skill set that
Yeah.
kind of singles you out and then enables you to do things. Yeah.
I mean, we sort of touched on a little bit where the intersection of computer science and biology has happened in a very similar way to physics and biology, in that bioinformatics just became a job that paid very, very well, and you didn't need a PhD for it a few years
Yeah.
ago.
Yeah. Yeah.
And saying to students, if you can code, particularly in Python, you can do an undergrad, maybe a master's that involves some bioinformatics, and then command a pretty decent salary as a bioinformatician. But you're right,
Yeah.
that's sort of where the landscape could. We don't know what that landscape looks like there going forwards.
It could. It is. It's really tricky. So one of the projects we're embarking on at the Centre for Computational Medicine is creating an AI-enabled lab where we cheaply try and automate a lot of the roles of sort of biological technical staff. Where so a lot of the so some of my students who are involved in doing the tissue culture work, they have to maybe come in at a weekend because they have to change the medium for their cells, which is the sort of the liquid surrounding the cells. They have to say, okay, let's take this one off, let's put some new stuff in, keep it all fresh, and keep any infections at bay, that kind of thing,
Hmm.
which is quite menial work. Is really important and significant, but do they need to be coming in at a weekend to do that? It kind of limits how many cells we can have running at any time. So we're sort of experimenting with automation, which isn't a new thing. We've got a robot arm that can take these things out, can draw up the liquid, and sort of change it over. And we thought, okay, AI. What can we do with AI? Because we know about AI and how to implement it. What if we had another robotic arm holding a microscope that could then check for how well the cells are growing, how quickly they're growing? Could we start getting cancer cells with certain mutations to compete against each other and then record the data automatically?
Wow.
Okay, you could probably do that. Well, okay, what about if we got something like ChatGPT to then direct the experiment to say, okay, so ChatGPT, you come up with a hypothesis, conduct the experiment to set the robotic arm off to do it, and then interpret results and then decide what to do next. And it turns out actually even ChatGPT can do it that
God.
fairly well, which means it's not only the sort of technical staff doing I say technical staff, it tends to be PhD students who are doing the sort of medium change stuff, but actually you can replace me potentially, the one that's kind of coming up with the hypotheses and trying to direct this stuff by an AI. And this is even, at the point of we don't have generalized intelligence. We just have these large language models, which don't really think in any kind of meaningful way. They just kind of generate text based on, what text has come before, but they can come up with hypotheses. Now, there's going to be a limit to that because it won't be a new hypothesis, I doubt. It's probably be something that someone else has reasoned before that they're just sort of replicating. So is it going to kind of create new science and new understandings? This is sort of the debate. So
Hmm.
hopefully there's still room for human scientists, but
Hopefully.
that may not be for long. And I on one hand, love that because suddenly, it could change the world in all sorts of ways that humans are limited by. We're limited by, our ability to do a lot of work and not get tired and,
Mm.
scale up. But equally, we're pushing us out of the picture, and, our sort of desire to have meaning and purpose in the world could easily be removed. So
Mm.
it's a tricky one, but we're pushing ahead with the pilot experiment anyway. It's really
Well,
difficult.
it's hoping it's disruptive in all the right ways. It sounds
Exactly,
amazing.
exactly. And I
Um,
wouldn't want to put off any students from, still getting into it, because I think there's still, of all the ways in which AI could be usefully disruptive in a good way, I think science is one of those.
yeah,
So yeah, it's definitely a good place to be, I think.
yeah, certainly. Just in wrapping up, what are the big frontiers or questions in your discipline? What comes first to mind?
Um, there are so many, and we it's interesting. I don't necessarily think I have a discipline. I'm trying to think what my discipline is, because
Or
it's not
biophysics in
physics.
general, then I suppose.
Biophysics in general. I think moving to interesting new models is a good one. So people are kind of less enthusiastic about using animal models
Yes.
and moving towards, in silico and in vitro in novel and interesting ways. We still need the animal models, but I think finding a way to kind of move away from those is a really key thing. And there's lots of interesting, exciting new thoughts about how to do that. Um, and that tends to be in vitro, but I think in silico really is the way forward, and kind of
Mm.
combining all of these things together in a way that's greater than the sum of the parts. And that's often difficult in maths. It's often the case with maths that what you put in is what you get. If you put two and you put two, you're always going to get four. But it's, sort of scaling it in a way that something emerges beyond what you've put in. That's the interesting thing. And that needs a lot of computing. So also the ability to scale up what we can do computationally and not emit a whole load of CO2 in the process from electricity
Yeah.
generation. So, doing this efficiently and green and sustainably is also a really important aspect.
Yeah, yeah, certainly. Absolutely. If people want to find out more about you and about your work and research, where should they go?
So we've got a page on the UCL website. So if you look up computational medicine on the UCL website, that should have an email address. Don't spam me, but send me an email. And, so we do summer internships often. And there's all sorts of interesting opportunities for doing courses and PhDs and that kind of thing. So yeah,
Mm.
look us up there.
Are you taking postdocs?
When we get funding too, we do. Yep, absolutely.
Right. Terrific.
Yeah,
Thank you so much. I know we could probably talk for another hour or so on this. It's amazing, but it's always great to catch up with you. Thanks for joining us today.
great. Thanks, Sam.
Thank you so much for watching. Don't forget to like and subscribe. Stay tuned for more amazing conversations with incredible researchers who are changing the world we live in and driving humanity forward. Feel free to check out my Instagram at polymath_Sam, or go to my website at www.sam-mckee.co.uk.